Everyone again to the Green Era Hub webinar series. My name is Barry Crosby and I'm one of the communication officers for the Green Era Hub project. Now, if you've been here with us before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERA-NETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here in October last year, and we've had 4 very successful series so far. The first one was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was Sustainable Food Systems, and the one that we just finished before the Christmas break was Precision Agriculture Technologies for a Sustainable Future. If you're interested in listening to the talks related to any of the topics from the series just mentioned, our recordings are now available to view on our YouTube channel. Now, for the next 3 months, we are focusing on the topic of plant health and protection, where we'll be hosting a series of expert-led discussions aimed at fostering a deeper understanding of plant pathology, integrated pest management, and sustainable agricultural practices. And just a few housekeeping announcements. Please do feel free to type your questions in the Q&A and chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. If at the end of the session you have a question for either of the speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Dr. Hugh Evans and Dr. Perrine Portier, and we'll open our session this morning with Dr. Evans, currently a research fellow at Forest Research in the UK, where he specialises in tree pest research and management. An expert in the field in pest risk analysis, Dr. Evans' work focuses on the threats posed by the global movement of pests through trade. Throughout his distinguished career, he has offered specialised knowledge on tree health with a strong emphasis on national and international phytosanitary and biosecurity issues. He's particularly focused on integrated pest management strategies, including the use of biological control agents, and has vast experience leading funded projects and publishing work in high-impact scientific journals. Today, Dr. Evans' talk is entitled Range Expansion in the Bark Beetle genus Ips, Findings from the Euphresco ECLIPSE project, which is part of the Euphresco Phytosanitary Research Initiative. Dr. Evans, thank you very much for accepting our invitation for our webinar series. I'm going to stop sharing my screen now, so you can start sharing your presentation. Thank you very much.
Thank you very much. Right, good morning, everyone, and thank you for that introduction. As was mentioned, this is just the outcome mainly based on an ECLIPSE project under the Euphresco banner. I was the project leader on that, and then we had partners from Belgium, Ireland, and the Netherlands and the Slovak Republic. So it's too many people to actually put on the screen for that, but hopefully you'll all enjoy that one. So there it is. Okay. So the main starting point is to ask the question, why do we worry about these damaging beetles? The genus Ips is worldwide, but there are 6 species in Europe, and they are a worry partially because they are so highly damaging. Almost everywhere they turn up, they kill vast numbers of trees. The other factor that we've looked at quite carefully, of course, is the fact they move worldwide, and I'll come back to that in terms of movement in trade. And there is increasing evidence of new locations being colonized, especially in the last 100 years or so. And so that's been the main focus of the project. So all of the six species, the European species, are rather similar in biology. I don't want to go into great detail on this, but the big one, shall we say, the most damaging of them all, Ips typographus, not very big, maximum size about 6 millimeters, so not very large, but they can have up to 3 generations a year. And we're always interested in the ratio of females to males because of the way the males attract the females, which I'll come back to. And the fecundity is not that great, only up to about 60 eggs per female. But of course, that can easily add up to bigger numbers. The main hosts are on the conifer genera, mostly spruce in Europe, but also pine and larch. So those are the basic biological characteristics. Should point out that Ips sexdentatus is the biggest of the beetles, up to 8 millimeters. So Ips typographus, we tend to talk about more than the others because it is by far the most damaging. And the strategy it has is that the male gets to a suitable tree, normally initially weakened or recently dead trees, because they have relatively little defense in terms of resin flow, etc. And then they will attract females and develop a so-called nuptial gallery. And along that gallery, you can see on the right here, the females lay their eggs, and then the larvae move outwards from the initial gallery system. And that forms a very characteristic pattern. And this is typical of different bark beetle species. We can tell the species from these patterns without necessarily having to see the adult beetle. And so you get that buildup on weakened trees. And then if they build up sufficient numbers, they move on to attack living trees. And very large numbers of beetles are required to kill a living tree, but these beetles can achieve those numbers. And we can see at the bottom some of the track record of massive area-wide tree mortality. Between 1850 and 2000 in Europe, about 8% of all the trees killed in the whole of Europe were due to this one species of beetle. Some big numbers from Switzerland, as you can see, 8 million cubic meters, 18 million cubic meters in Austria in that short period. And these equate to millions of trees being killed. So we do concern ourselves with this particular problem. And these photographs sort of illustrate the combination of biology that goes on to result in tree mortality. This photo on the right in the bottom, you can see the lying trees that were probably from a wind throw or even felled and left there. And the beetles like these trees because they're weakened, and therefore they can build up in them and then move on to the living standing trees, and they've been killed as well. And you can see this patch dynamics. The beetles start from small foci and then gradually build up. And as you can imagine, if there are 3 generations a year, this buildup moves on very rapidly indeed. I've mentioned they move internationally, and all over the world they've been intercepted. These are the European species, all intercepted in the USA, for example. As far away as New Zealand, they've also intercepted them. And in the UK, of course, and as indeed for Ireland, we tend to think of ourselves as being slightly protected by being islands. But we get this incursions of beetles on imported wood. And just post the Second World War, there were hundreds of beetles brought in on logs from Germany, but they never established. And we've also done pheromone trapping at the ports. There's a very effective pheromone for Ips typographus. And we've had captures over many years. You can see some of these numbers were quite relatively big in the early 90s. And so we looked at expansion of the range in our ECLIPSE project. And one of the analysis that we carried out was mapping over time the records of Ips typographus all over Europe. And what we're interested in particularly is the blue areas, the blue colors, because this is range expansion. Back in 1900 or so, you can see the red and yellow dots. the range wasn't all that big, and it mirrored the spruce population at the time. And the similar story for Ips exdentatus, one of the pine attacking species, mostly moved down south in this case, rather than widely as Ips typographus has done. And so in the Eclipse project, we've actually published a review paper on this whole story just before Christmas, actually. And so that can be found in Entomologia Generalis, and the lead author was Jean-Claude Gregoire, one of my colleagues in Belgium. And you can see some of the analysis here that on the left maps, we've got the orange areas, which is the expansion in range of the, oh, sorry, this has gone back, of the host tree species. And you can see that the orange patches having increased enormously in the last 100 years or so. And then on the right, you can see the range of the particular species. We got 3 species here, Ips typographus, exdentatus, and amitinus. And these are all relatively damaging beetles, and they've all essentially tracked the availability of their host tree species. And you might say, well, you know, that was obvious, wasn't it? But in fact, it didn't happen that quickly. And we've been wondering why sometimes this range expansion didn't track the tree expansion as quickly as we first thought. So how do they keep up with this? Well, we mentioned trade already, and they can jump spread, shall we say, but that tends not to result in great infestation levels, because the numbers arriving tend to be relatively small, and they then disperse, and they don't necessarily find the host trees. So we look at now more at adults themselves flying or being blown long distances. Mostly they fly locally, up to about 1 kilometer. But we've now got evidence that they will fly or get carried on wind very long distances. In the north, in Finland, they found the beetle at least 43 kilometers away from the nearest spruce. And we've now recently been able to study cross-channel flights of beetles arriving in Kent from France and Belgium. So going back to Scandinavia, again, we've covered this in our recent review paper. Some of the 3 species you can see, Ips typographus, amitinus, and sembre, they've all expanded their ranges with the expansion of conifers in Scandinavia. You tend to think of those countries as already having a lot of trees, but they've also expanded the numbers of trees. But in this map, we can also see here lots of records of the beetles way outside the actual known tree range. And this, again, reflects long-distance movement in the air or in flight. And in more detail, we've looked at the recent incursion of Ips typographus, who's been found for the first time breeding in Kent on very weakened Norway spruce trees. And so we carried out a very large study. My colleague, Dagan Inward, again with Jean-Claude Gregoire, has looked at a whole range of pheromone traps, a sort of transect from the Ardennes right into Kent. And these graphs essentially confirm that these beetles move along a sort of transect, a density transect, from these very high populations over here down to lower but still significant populations in England. And we've estimated from modeling this spread that they should have reached at least 160 kilometers inland. So this is remarkable long distance dispersal. And so our concern about the channel ports being the main points of entry on trade perhaps are a little overblown, because we're now finding the beetles don't bother with that and fly over themselves. And then we've looked in more detail at the numbers of beetles in the southeast, and you can see an awful lot near the channel itself, but way inland. And this confirms the distances that the model was suggesting. So what's going to happen? Are these beetles going to establish Well, we did think ourselves, possibly our climate might not be suitable for establishment, but a recent study by Kerrion Webb and colleagues in Cambridge University have done a bit of modeling, and they've adapted a European model from Austria called PhenIPS, which is the phenology of IPS attack. And their estimate is that 2 generations, at least in the southeast, and even in the far north, we now think they might manage 1 generation a year. So in fact, there's no climatic barrier to IPS typographus making its way through the whole of the UK, and indeed, if it manages to get to Ireland. But of course, suitability of host trees, etc., might limit that. So what do we do about managing these things? Well, I mentioned already that damaged and fallen trees are the main starter points of these. And so if you can manage and remove that type of material quickly, then you can also remove some of the beetle risk and some of the beetles themselves. But you've got to move fast, because when you've got 2 or even 3 generations a year, you have very little time to cope with the beetle numbers. And trap trees can be used, and then remove those trap trees, but again, an expensive and slow option. And in Finland, they have a policy of removing more than 20 damaged conifers to try and prevent further buildup. So natural enemies, we're always interested in the possibility of biological control. There are big numbers of natural enemies, but all of these, there's a range here from parasitoids through to the different types of predators. They're all generalists, and that's one of their drawbacks, in as much as they tend to react only when beetle numbers are very large, the pest numbers, and none of them are host specific, unlike the one that works very well for Dendroctonus micans, another bark beetle. So they can give you, and some data here from Beat Vermelinger in Austria, in Switzerland, I mean, the numbers can actually give you up to about 80% mortality of Ips typographus, but 80% kill of multi-millions still leaves you with millions, and that's the problem. So managing the proportion of host trees, we're also finding is another factor. We've found this in our study, and others have done a lot of work on this. And in Sweden, for example, volume of tree is a factor. Increasing tree height is a factor. And the fact that people harvest wood and perhaps don't take the wood away quickly enough means you get local buildups, and they can actually increase populations locally. But one of the interesting things is that if there's more than 25% spruce of birch in a stand, then you get far fewer beetle attacks. And in Austria, they're trying to do that same thing, decrease the proportion of spruce, shorten the rotation length so the trees don't get too big, and move timber quickly. So those are all what we call slow drivers, but they require a lot of management commitment. And likewise, climate effects are becoming increasingly important because you get higher temperatures and increased storms. And this is one of the big factors, is wind throw. Climate change is causing a lot more wind damage, and those are outbreak initiators, as I mentioned already. And then you can predict the foci of attack based on wind damage likelihood, and there are various models you can use for that. And so then you start looking within those windbreaks for signs of attack and manage those starter populations if you can. But as I mentioned earlier, this is very labor intensive and requires a heavy commitment. And so slowing the spread of infestation requires this intense local management. And this is what's happening in Britain at the moment. We're trying to slow the spread of this newly established Ips typographus attack. But as I've said already, this is labor intensive, and in more remote areas, very difficult to implement. So on that slightly depressing note, I have to say that managing these pests continue to be a very major issue in Europe. And some of the knowledge we've now got might help increase that in terms of future forest management. And so just to end with a few information sources, you can look at what we're doing in forest research, the Euphresco project I've just described, the major review from that project, and another review that I produced a couple of years ago, which is PDF format, which is downloadable from our website. So thank you very much.
Dr. Evans, thank you very much for such an interesting and insightful presentation. I think just so many fascinating aspects about it. I just want to remind everyone that the chat box and Q&A section is now open. So please do feel free to type a question for Dr. Evans at any point. Dr. Evans, just while we're waiting for the questions to come in, I was just really curious about, naturally, I suppose, Ireland being the host of this webinar,
Yeah.
about the spread to Ireland. Like, given the short geographical distances, relatively speaking, that the Ips bark beetles would have to cover, I find it amazing that there's been no kind of identification of Ips bark beetles in Ireland so far.
That's correct. We did have Irish partners, in fact, both from the North and the Republic. And yes, it's a big worry. I think what our main concern now is that Ips typographers particularly, but all the other Ipses as well, don't get to the main spruce areas of Great Britain. And those main spruce areas are in Wales and Scotland. And then you're that much closer to possibilities of flight across, let alone trade. We manage the trade with Ireland very effectively in terms of debarking, etc., etc. A lot of inspection goes on. And that applies particularly to Ips sembrae, funnily enough, which I didn't mention a lot there, the larch bark beetle, because that is present in Scotland. And there are very strict rules about exporting larch from Scotland to Ireland. But of course, this has now thrown up that extra worry that you touched on, that they could actually fly over if big enough numbers can build up either in Wales or in Scotland.
Fascinating stuff. Thank you very much. Just a question from our audience, from Alan McLeod. He's asking, what is the next bark beetle the UK or Ireland should be worried about?
Oh, gosh. I would say perhaps isn't that list big enough already, you know? But in fact, we do worry a lot about North American bark beetles. We do import a lot of wood from Canada and the US, and they have a whole range of other Ips species, but particularly the Dendroctonus species. These are very similar bark beetles, very similar biologies. And particularly mountain pine beetle, which is in the Pacific Northwest, is an absolute tree hammering beetle. They've had massive outbreaks there. So we concern ourselves very much with that. And again, we try and manage those threats through both trade of wood as a wood product, but also particularly wood packaging material, which is where most of these incursions come through. And there's a new rule called ISPM 15, which requires that all wood packaging material is bark-free and or heat-treated. So I would say North America is our next biggest threat.
Thank you very much. Just another, well, a comment really, just from a member of our audience. They say increasing the proportion of Abies alba in Norway's spruce forest would help to increase resistance. Would that be something you would agree with?
That's possible, yes, indeed. I think the key thing there is the mixture side of things. Obviously, it's better if you have a non-conifer host among your mixtures, but Abies alba, white spruce, white fir is a possibility because very few beetles seem to go on that, and so that's a good suggestion.
And just another question coming in there, Dr. Evans. One of our audience says, thanks for the great talk. What is the best way to access the FENIPS style maps of the British Isles that you referenced, if there is any way?
Well, I think the key is download the paper that I referenced there, which is actually in open source, so it's free to download. And so I don't know if they can access the link that I put in my final slide, but that's the best way to have a look at them. Um, and, and, and then the paper that I mentioned with, I'm sorry, the FENIPS maps, which are the climate ones, that's in also in a open source paper from Kieran Webb, Max Blake, and Christopher Gilligan. And that was published in, what was that? Plants, People, and Planet. So that's open source. I can type that in shortly.
Yeah, Dr. Evans, I mean, if it would be agreeable to you, I could suggest that the audience member who was asking that question could maybe contact me via email, and
Oh,
then
yes,
I could
and
either
I'd
put
be
them in
happy.
touch with you, or if you want
Indeed,
to send
yes,
me the information
absolutely
and I
happy
can
to
send
do
that
that.
on.
Yeah,
Yeah,
got it.
no problem.
Okay.
Thank you very much. I was just another kind of question that I was really kind of curious about was the kind of you touched on climate and you're kind of touching on the kind of natural changes to forest structure. I was just wondering about human activity, the role of humans in this, and I was wondering how do human activities such as logging or land use changes interact with the ecological factors affecting the increasing numbers of
Well,
Ips bark beetles?
yeah. Well, I would say probably based on this study we've done, and obviously lots of other literature, the human factor is paramount, because essentially that big change in the range of the host tree species is all human driven. That's not natural spread of the trees. That's afforestation, both for commercial and for landscaping and other reasons. And so by humans giving the beetles their resources, they're actually creating the problem as well. And also changes of management. It's become more difficult and more resource driven to actually manage these outbreaks by rapid removal. We used to have a thing in Britain way back called the 6-week rule. And essentially, if you get an issue of a bark beetle, if you get everything off site within 6 weeks, then you take the problem away. You literally do. And after the 1987 hurricane we had in the southeast, there was massive, massive amounts of pine on the ground, and we got huge infestations of both Ips sexdentatus, the pine Ips species, and a thing called Tomicus spiniperda, which is a shoot beetle. And trying to manage those was an absolute nightmare because they had a massive resource brought in by wind and way beyond the resource availability to remove the trees.
That's fascinating stuff. Dr. Evans, thank you so much. I mean, I think we could go on for another
Yes,
half an
well,
hour.
it's always a
But
pleasure to talk about it.
it's an absolute pleasure to listen to you, and I mean, just a fantastic presentation, such an interesting and really scientifically worthwhile project. So thank you very much for that. So now joining us is Dr. Perrine Portier, who is currently research engineer at INRAE in France, where she also serves as director for the French Collection for Plant-Associated Bacteria. A leading expert in the management and preservation of bacterial resources for plant protection, Dr. Portier's work focuses on the phylogeny, taxonomy, and pathology of plant-associated bacteria, as well as the epidemiology and diagnostics of plant pathogens. Dr. Portier's work contributes significantly to advancing plant protection throughout the development of molecule diagnostic tools and the management of plant-associated bacteria collections. Throughout her career, she has been dedicated to the distribution, preservation, valorization of strategic bacteria resources for the global scientific community. Today, Dr. Portier's talk is entitled Building a Reference Database for Plant-Pathogenic Bacteria Identification by MALDI-TOF and Evaluation of the Method, which is also part of the Euphresco Phytosanitary Research Initiative. Dr. Portier, thank you very much also for accepting our invitation for the webinar series. We're absolutely delighted to have you join us today. So without further ado, the floor is yours. I'm going to stop sharing my screen now so you can start sharing yours. Thank you very much.
Thank you for the invitation. So today I will talk about the MALDI-TOF technique. And without further ado, okay, well, the MALDI-TOF technique stands for the matrix-assisted laser desorption ionization time of flight. So this is a technique which is already in use in a lot of different areas for microorganisms identification. And it works, you put your bacteria or your fungi, because it works, it may work also on fungus, but I don't have a lot of information about that. You put your bacteria within a matrix, and then you put a laser beam on that. This will have as an effect to vaporize all the proteins inside the bacterium, and the proteins will be charged and will fly away in a hard vacuum tube. And then they will go down after a while, and they will be separated by their weight. In the end, you will obtain a mass spectra. I will take that. Okay. You will obtain a mass spectra. And if you have the opportunity to compare it with a database of the reference spectra, you can have the identification of your bacteria. As I said, it's already widely used in human medicine because the spectra can be pretty specific to one taxa or another. But in the end, actually, the database of the reference spectra is critical, and especially the quality and the accuracy of the database may drive the accuracy and quality of the identification. Though for human medicine it works, but how for plant pathogenic bacteria? How does it work? So there had been already a lot of different initiatives to test this technique for several genera like Burkholderia, Erwinia, and so on. But actually for plant pathogenic bacteria, there were no general database and no general evaluation of the method. And that's what we did during this MALDI project. And it's for the rapid identification of plant health-related bacteria by MALDI-TOF mass spectrometry. And the objectives of the project were to create a reliable and complete reference spectra database for at least some of the genera of plant pathogenic bacteria. And after building this database, the idea was to evaluate it, the method, its applications, its limits as a diagnostic tools. We were several partners involved in the project. I think our project was much smaller than the one Dr. Evans presented. However, we were 5 institutes representative. And why MALDI-TOF also, I forgot to mention that it's like magic, the method, because it works in like half an hour. You took your bacteria, you put it on the specific plate, you put it in the machine, and half an hour later, you have your results. You know what it is. I mean, that's just a dream for bacteriologists, and especially us as a collection. Anyway, to evaluate the method, we needed to have access to reliable biological resources. So these resources were available from the partners, and especially my lab, because, as you said, I'm the curator of the French collection for plant-associated bacteria. In the collection, we have more than 7,000 strains today of bacteria. Our big strength is that we have representative of all the known diversity of plant pathogenic bacteria. We do not have only reference strains. And for instance, we have more than 2,000 Pseudomonas, almost 2,000 Xanthomonas. We have one of the biggest collection of Xylella, public collection of Xylella in the world, like 400 Ralstonia, and so on and so on. We have these strains preserved following 3 preservation modes, either freeze-dry, in vials or in cold, either in liquid nitrogen or in a freezer at -80. Sorry. And these strains are available for the world scientific community, and we distribute that to the scientific who need them. So if you want some more information, please visit our website. And our characteristic also is that on the world collection, we performed molecular characterization, meaning that for all the accession, we sequenced 2 housekeeping genes. And this made us experts in the diversity of these bacteria, but also we have reliable taxonomy for most of the accessions, and we know precisely who these strains are. And that was critical in this project because we needed strains already well identified and well characterized. And to build this reference database, we based first on reference strains. We took the type strains of species and subspecies and part of ours. And we took also other representative strains of the different taxa based on our knowledge of the diversity of each taxa. So we decided first to focus on Pseudomonas syringae, Pentoria, Curtobacterium, Pectobacterium, Xylophilus, and in a lesser extent to Erwinia, Xanthomonas, Clavibacter, and Ralstonia. We also standardized the culture conditions because MALDI-TOF actually the method looks at the proteins inside the bacteria. And doing so, it's sensitive to the phenotype. And following, if you cultivate your bacterium on a low or on a poor or rich medium, the proteins produced may be not the same, and that may alter the spectra later. So along with that, we decided to stay focused on a few media. And if you want to analyze one bacteria or the other, it's better if you can use the same culture conditions as we did. And then we did produce the reference spectra. We took our bacteria, we put them in the machine, and we asked the machine to produce a reference spectra from these specific strains. Today in the market, there is 2 main systems for the MALDI-TOF analysis and identification of microorganisms. A system by bioMérieux and another one by Bruker. Both include ready-to-use databases. We decided to work on the Bruker system. The first reason was that all the partners had access to a Bruker system, and only one of us had access to a bioMérieux systems. So that was a really good reason, actually, to do that. The other reason is that the Bruker system is a bit more flexible in the way that it's easier with this system to create your custom database. And when you want to analyze a strain, you can either analyze it on the Bruker database or on your own database or on both databases simultaneously. And after building the database, we decided to analyze strains, and we analyzed again the same strains that we used for the MSP database, the main spectral profile, in order to check if it works. And we also work on other representative strains from the considered taxa in order to be able to compare between the obtained and expected results. So I'm based in Angers in France, and all the MALDI-TOF analyses were made in another location in a platform named Pixanim, which is located 150 kilometers away from our lab. And the results. Okay, when you receive a MALDI-TOF result, it looks like that. So here you have your sample and the first match and a score value, the second match and a score value. If it's above 2, it's nice. If it's between 1.7 and 2, well, it's not so good. And above that, it's not good at all. And what we did, for instance, that's the result for the Pantoea taxa. So we decided to analyze, we did that in duplicates every time, some strains, this one from the Cypripede species, and the best match were also for the Pantoea cypripede spectra, perfect. Everything was fine, good identification for genus and species. However, we found out that at subspecial levels, it was variable. It means that sometimes the identification was perfect and sometimes it was not. And it's pretty hard to determine why it was good and why it was not good. Into the results given by the MALDI-TOF, it was not possible to determine if the results were reliable or not, actually. And we also put in the machine some strains. We knew we had identification problems on that, just to check. And in fact, the method can correctly identify that there were some problems. So for this taxa, the identification is correct at the species level, but not always at the subspecies level. And then we move to Pseudomonas syringae, the syringae lineage only. Pseudomonas is a huge taxa, a huge genera, but we focused on the syringae lineage. And Gambilla and collaborators in 2017 published a phylogeny of these groups, stating that in the Pseudomonas syringae, there is certainly at least 6 phylogenetic groups. And inside each group, there is probably more than one species. And inside each species, there is some pathogens, meaning that really closely related strains can have highly different host range. And the disease provoked by these strains may be totally different. So the management behind that maybe not the same. So it's pretty important to know what you're talking about and what you have in your crops. So we decided to create a high number of reference spectra for each group of this syringae lineage. And then for each group, we analyzed even more strains to be able to compare if the method was working. Okay, so in all cases, it was possible to identify that these strains were indeed belonging to Pseudomonas. But as soon as we gone down to the phylogenetic groups defined by Gambilla, the accuracy was less good. And even when we were going down to the species level, it was even less good. And I'm not even talking about the pathogen subdivision, because in that case, the method was not able to discriminate between strains. So what does it mean for plant pathologists? As a general conclusion, we can say that the MALDI-TOF identification is correct at the genus level with our database in all cases. But for the species level, it depends. It depends on the taxa you're looking at. For Pseudomonas and Pectobacterium, which are known to have pretty closely related species, it doesn't work really well. In other taxa, it works better. Curtobacterium is in yellow here because we did the work before the Curtobacterium taxa species was divided into different species. So maybe we should reanalyze that in another way. And if you look at the subspecies level, it's often not reliable enough for that. But in plant pathology, the relevant level, the relevant taxonomic level for the diagnostic is often at the intraspecific level. For instance, Panthera stewarti, subspecies stewarti, is classified as quarantine, meaning that you cannot export and trade seeds having this bacteria in them. But Pseudomonas, sorry, Panthera stewarti patova, subspecies indologenes, which is really closely related, is not quarantine organisms. So if you make a mistake in the diagnostic, you can throw away tons of seeds, which could be good actually. So it's a huge, this kind of misdiagnostic can have a lot of impact. However, this MALDI-TOF technique is actually a very useful tool for fast screening because it's pretty fast. And if you know already if your bacterium, it's a Pantoria or if it's a Pseudomonas, you won't act in the same way. So this can bring you very useful insights. And for crop management, only having the genera of the bacterium you have, sometimes it's enough, sometimes it's not. But if it's not, you will be able to be much better at defining what's your problem because it can guide further inquiries. So MALDI-TOF alone is not enough for use in diagnostic, but it can be a really powerful tool for that. And we found out that it's more or less equivalent to a 16S sequencing. And it's already in use in different collections as a replacement of the 16S sequencing because it's much faster. In our collection, it doesn't answer the question we're talking to us. So we decided to not go further with the MALDI-TOF technique for these reasons. However, I'm really happy to have done this work so that it can help other scientists. And now what? So the outcomes of this project were the spectra database we built, which is now fully available through the web. You can download the bundle and put that in your broker system, and you can use our database to identify your own strains. That's no problem for that. And the evaluation of the method will lead to an EPO standard. I hope the publication will occur in 2025, maybe next year, I don't know. It can take a lot of time to that. And after that, perspectives, that's beyond our projects. Now we have a huge, much bigger project. Well, I won't lead it, and I will be only a small part on that. But the MALDI-Bank project, which is financed by the European Union, actually it starts in 2 weeks, in February 2025. For 5 years, it implies 24 partners, and the aim of the project is to build some tools and a community, to having a community generally shared database, which would be implemented by everybody, more or less like an NCBI for MALDI-TOF spectra, and not focused on plant pathology, but on all the thematics you can imagine. And I think the biggest part of this database rapidly will be about human health. But of course, the plant health has also its role to play in that. And I think it can be a really good outcome as a collective initiative. And now I thank you for your attention. I just want to point out that this work, the evaluation of the method, was possible because we had the collection behind. Other works in determining the usefulness of other diagnostic tools were possible because we have the collection. But the collection, they are critical. They are really a good tool for research, but they are only as good as what's inside. So please, when you're isolating some interesting strains, consider to deposit them in collection to make them available, because over the time, we would be able to have a really good insight of epidemiology, for instance, or to just monitor what it is and to have always up-to-date tools for what it is in the nature. So deposit in collection is an investment for the future. And thank you again for your attention.
Dr. Portier, thank you very much. Again, another fascinating talk. I really, really enjoyed that. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Dr. Portier at any point. Dr. Portier, just while we're waiting for a couple of questions to come in, I think you may have answered my question just with your last slide, but I was just really fascinated about the selection criteria for MALDI-TOF, like particularly in relation to like the bacterial strains chosen. And I was just wondering, how do you select the different bacterial strains to be included in the reference database?
Okay. We first focus on the genera, which were, of course, of an interest for plant pathogenic, for plant pathology. And we also focused on the genera where we had the most of data, identification data, because it was important to be able to compare the MALDI-TOF diagnostic tool to other reference tools, actually. So we choose the taxa where we had the best results at the point when we did the project. And then inside each taxa, because of the housekeeping gene sequencing we made, we had the whole phylogenetic trees. And we were able to understand the diversity within this taxa. So we focus on strains. Well, it was a bit arbitrary in a way, but on a phylogenetic tree, we took the type strain and strains being not too different and also the most different as possible. The idea was to be able to know if we could cover the whole diversity. And these criteria were the good one for this project. I don't want to say that in each, following your scientific question, maybe the criteria will be different. Mm-hmm. And that's also the power of the collection, because as a scientist, you can go to the collection curators and ask them, okay, I want to do that. Which strain would you recommend to answer this particular question? And we can work with you to find out the best criteria for choosing the strains you need, actually.
Sure. Very good. Thank you. I mean, just again, kind of following on, I suppose, from that question, one of the kind of, I suppose, challenges in creating a database of anything is the issue of keeping that said database up to date. So I was just, and I know you kind of touched on this a little bit in your presentation. I was just wondering, can you maybe tell us a little bit about how you ensure that the MALDI-TOF reference database remains up to date, particularly in relation to like emerging pathogens, for example?
Yeah, that's a good, I mean, that's my whole life, actually.
Yeah.
So you're designing something and like 1 year after, or even further, even faster, it's out of date. So that's a huge work, actually. And I think that can be done only collectively, because the collections, also the Biological Resource Center, are the collective results of the collective work, actually. The strains I have in my collection are from the oldest were isolated in 1804, something like that. And they come from all over the world. And I isolated, well, maybe 1 or 2 of them over the more than 7,000. But because of this collective work, they are all in the same place, and they are useful in that case. And the updating of the database, of course, it's an ongoing process. It means that we need regularly to check the reliability of the database, and from time to time to identify strains with the MALDI-TOF, and also to check also with other means. And if at one point we saw a discrepancy, we need to add the new spectra into the database. That's why I think the MALDI-Bank project, which starts in a few weeks, will be a real bonus for the whole community, because the MALDI-TOF hasn't really spread the use outside the hospitals and human clinics because of that, because the databases were scattered all over the place. And even in the hospital, it works better for the human pathogen, because the population structure of the human pathogens and the environmental pathogen we have is different. In the human pathogen fields, the pathogens are almost clonal to each other. They are really alike. So you have really discrete groups of pathogens. But in the environment, you have a whole continuum. that's why the MALDI-TOF is not as good to discriminate between them as in the human field. So in the human field, they always put their new spectra in their databases, and we need to do the same for the plant pathology. But we are not organized in the same way. We're talking about plants which are all over the place and not disease. People are all in their hospital. It's easier to have access actually to discriminate between them. So this will need a huge work. And the MALDI-TOF project, the aim is to have a tool to implement your own spectra with time. So every time you will be able to do that, you can, in the end. So maybe in 5 years, we'll have
At the answer, absolutely. Well,
product.
I think it's fantastic to actually hear about the MALDI-Bank initiative, and a really good sign that the first project using MALDI-TOF was very successful, and leading on to this 5-year project. It's really
Well,
encouraging indeed, I
actually,
think.
I'm not in the initiative of the MALDI-Bank. I mean,
I
it's
see.
not the human sector,
Yes.
but still being part of this MIRI infrastructure, for instance, and the whole community of collections, we said, okay, we need to do something there.
Yes.
And you're right, the MALDI project showed that it was useful to do that.
Yeah.
And
Absolutely.
our data, our spectra will join the MALDI-Bank database. For now, there is no website yet, but soon it will come, I'm sure.
Fantastic. Dr. Purdy, just one last question. I was just wondering about how the accuracy of MALDI-TOF identification compares to more kind of traditional methods of identification.
Well, it depends which method you're talking about. The MALDI-TOF, as I said, is more or less like the 16S identification. In bacteria, we know that 16S is reliable to identify the genera. And following the taxa you're looking at, it can go further or not. It depends. But sure enough, the MALDI-TOF will have difficulties to discriminate between different strains and between different subspecies groups, which are closely related. It can be a big, wonderful tool or not the one unit, but it's the same for other techniques. For the PCR tests, taxa-specific PCR tests, some of them are really useful, some of them are not useful. It depends what you're looking at. That's always the difficulty.
Very good. Dr. Portier, listen, thank you so much again. I think just like Dr. Evans' talk, I think we could be here for another half an hour easily, just discussing your research project. I'd just like to say, unfortunately, it's just 10 o'clock, so we have to wrap up, unfortunately. But thank you very much, everybody, for attending today. And of course, I'd like to express my thanks to Dr. Hugh Evans and Dr. Perrine Portier for giving us wonderful and insightful presentations, I think, and giving us such kind of great overviews of their respective projects this morning. Our next webinar will be on February the 6th, the same time, 10 a.m. Central European Time or 9 a.m. Ireland UK time, when we will continue our new webinar series on plant health and protection. I'll be sending the invitation out to all of you in the following days. Thank you very much again to our panelists, and thanks everyone for attending and for your questions. We really hope you enjoyed the webinar again this morning, and we hope to see everybody back with us in 2 weeks' time. So thank you very much, everybody, and have a great day.